Uh, Good evening. So we'll start today's session. Today, I will be summarizing the nine's content, which covered circulatory system and musculoskeletal system rather, and then we will look at few questions. Right. So I will start sharing the screen. All right. So in general, if we talk about circulatory system, it is a transport network found in most animals uh, as well as plants, responsible for the movement, uh, plants as in the vascular system in plants, which helps in circulation of water and food materials. But however, this week's content focuses on the animal's circulatory part. So in general, if we focus mainly on animals, most of the animals, circulatory system is responsible for the movement of essential substances like nutrients, gases, hormones, waste products, even neurotransmitters for you know uh, transduction, like signal transduction and all, throughout the body. While the complexity of circulatory system can vary across species, however, the fundamental function actually remains the same, which is to ensure that every cell receives what it needs to survive and function. Now, there can be majorly two types of circulatory systems. One is open and the other one is closed. So, two main types. The open one, what happens in the open one is the blood or a fluid called hemolymph is not always contained within vessels or tubules. Okay, it is pumped by a heart into a body cavity called a hemocoel. Seal means cavity. Okay, where it directly bathes the organs and tissues. And then this particular fluid re-enters the heart through openings called ostia. Uh, these osteopores we saw in the porphyrids, right? Sponges. So the fluid then re-enters the heart through openings, and this system is less efficient and is typical in invertebrates like insects, spiders, and most mollusks. In case of sponges, also you saw that it had a main osculum or mouth like as in the pore opening through which water can gush inside, and there were other openings on the body of the sponges through which again like tube like openings on the body of the sponges as well. However, if we are talking about the heart where and the hemocoel, this type of thing is found in invertebrates like insects, spiders, and many mollusks. And then when we talk about closed circulatory system, where the blood is continuously confined within a network of blood vessels, and a heart pumps blood through arteries which branch into capillaries, and then we will look at each of these terms. And then these are basically different tubes or tubules of varying diameters and thickness. And then this blood can flow back to the heart via veins. This allows for more efficient and rapid transport and even control of pressure as well. The system is found in vertebrates, including us humans, some invertebrates like earthworms, and cephalopods like squids and octopuses. Now, if we talk about the circulatory adaptations in different organisms, so different kinds of organisms have evolved using circulatory adaptations. So if we talk about sponges and cnidarians, they actually lack a true circulatory system. However, all these exchanges can occur via diffusion. Then there are annelids like earthworms. They have a closed system with multiple hearts as in some kind of pumping organ. And insects have an open system with dorsal vessels and hemolymph, the fluid which is found in the body cavity. And fishes exhibit single circulation with gill-based oxygenation. Then come amphibians and reptiles, which show partial separation of blood. They do not have complete four-chambered hearts. Then we have birds and mammals, which have a double circulatory system for efficient oxygen supply. So these adaptations reflect the evolutionary specialization based on the different kinds of complexities, their needs to use different kinds of resources, how their bodies are, designed, and definitely the metabolic demand. So if we talk about heart and circulatory organs, then heart is a muscular pumping organ found in most animals with a closed circulatory system. In fishes, the heart is two-chambered, having one atria or one auricle and one ventricle. In amphibians, it is three-chambered, two atria or two auricles and one ventricle. In reptiles, it is three or partially four-chambered, whereas in birds and mammals, it is a double circulatory system. It is four-chambered, allowing complete separation of oxygenated and deoxygenated blood. In simpler animals like insects, the heart is a tubular structure that pumps the hemolymph or the fluid through the body cavity. Now, vertebrates exhibit different patterns of circulation that correspond to their heart structure. Single circulation is found in fishes, where the blood passes through the heart once during a complete circuit, and the two-chambered heart pumps deoxygenated blood to the gills, where it gets oxygenated and then it flows directly to the rest of the body before returning to the heart. And when we talk about double circulation, what happens is it is mostly found in amphibians, reptiles, birds, and mammals, where the blood passes through the heart twice in one complete circuit. Okay. And this system is definitely more efficient because it separates the circulation to the lungs from that to the body tissues. Again, there are two loops. We talked that the blood can pass through the heart twice in one complete circuit. So if there are two loops, one is the pulmonary circulation and another is systemic circulation. So in pulmonary circulation, what happens? The loop that transport the blood between the heart and the lungs for gas exchange, and systemic circulation represents the loop that transports blood between the heart and the rest of the body. Okay. Now, when we talk about the components of different kinds of the constituents of the circulatory system, heart, the muscular organ that acts as the pump, generating enough pressure to move the circulatory fluid. The circulatory fluid can be blood or hemolymph. Okay. The structure of the heart can vary. For instance, fishes have a two-chambered heart, amphibians and most reptiles have a three-chambered heart, and birds and mammals have a four-chambered heart. Circulatory fluid, this is the medium that transports substances. Then if we talk about blood, it is a specialized fluid connective tissue in closed systems consisting of plasma and blood cells. And like blood cells, like red blood cells, white blood cells, and platelets, hemolymph, the fluid in open systems which combines the functions of blood and interstitial fluid. Blood vessels, the tubes that carry blood in a closed system. Then there are arteries. Uh, Again, these come under the group blood vessels. Arteries, which carry blood away from the heart. Veins carry blood back to the heart, and capillaries are very tiny vessels where the exchange of gases, nutrients, and waste occurs between the blood and tissues. Now, if we talk about arteries, we see here it is a thick-walled uh, tube or vessel, blood vessel, which is lined by smooth muscles. Okay, and arterioles are branches from the artery. Okay, so these are branching tubules originating from the arteries, and arterioles can actually constrict and direct the blood flow and even control the blood flow. Okay, they can increase or decrease the blood supply to the skin, suppose, as an example. Okay. Now, when we talk about capillaries, okay, these are much thinner as compared to arteries and arterioles, and body tissues contain a very vast network of very thin capillaries. So capillary walls are only one cell thick, allowing the exchange of gases, nutrients, and waste materials. And even if like capillaries are so fine that RBCs actually have to really line up in a single line to go through them. Okay. And when we talk about veins, so veins have thinner walls as compared to arteries, And like we looked at arteries are thick walled and arteries are lined by smooth muscles. Okay. However, veins have fewer smooth muscles, but veins do have valves to control the direction of the blood flow as well as pressure of the blood flow. Okay. Yeah. So there, these are the valves, and like based on whether the muscle is relaxed or contracted, based on that, the blood flow, I mean, the valves can open or close eventually, allowing controlling the flow of blood through the veins. Then comes venules, which are again kind of branching out of uh, the veins. Okay, they are smaller than veins, but however, they are still larger than the capillaries. Okay, and when we talk about blood velocity in venules, blood velocity actually slightly increases as the blood flows from capillaries to the venules because capillaries are really thin, like extremely thin. We looked at it like it is one, the wall of capillaries are like one cell thick. Okay, and when blood passes through such thinner portions and suddenly it gets to, you know, um, suddenly it gets a thicker space to flow inside, then eventually the blood velocity or the speed with which the blood enters the venules from the capillaries will increase. Okay, so this is what has been shown here, that slowly as the blood passes from the arteries to arterioles, uh, there is slight decrease in the velocity of the blood. However, when it enters the capillaries, which are really thin, where the blood velocity, speed of flow of blood decreases because you can see that these are so thin that even the RBCs, the single RBCs, have to line up in single line. Okay, one by one they are passing through. It's not that uh, like all the cells are randomly gushing in through the capillaries. Okay. And then eventually again when it enters the venules and eventually veins, which are thicker than venules, then slowly the blood pressure increases. Blood pressure as well as the blood velocity increases a little. Okay. And this is again the exchange that goes on between the arteries, arterioles, capillaries, but this is the intensive network of capillaries. Then eventually there are venules and veins. This interchange is shown here. And the types of circulatory system, the same things have been now represented pictorially, where fishes have one heart, two-chambered heart, where one atrium or one auricle and one ventricle is there. It is a single circulation where gill-based oxygenation takes place. Okay, and one round of exchange takes place at the capillaries inside the gill and then gills, and then the other ones takes place at the network present in the rest of the body. And then there's reptiles and amphibians where they possess three-chambered heart with two auricles, this one and this one here, and one ventricle, where both so eventually in three-chambered heart, both the oxygenated and deoxygenated blood get mixed. Okay. Then we have birds and mammalian heart, where complete double circulation takes place and the blood actually passes through the heart twice, and there are two auricles and two ventricles, nicely separated, four chambered heart. Okay, so if we talk about just kind of summarizing what are the key functions of a circulatory system, nutrient and oxygen delivery transports oxygen and nutrients to cells, muscles and organs of the body. Waste removal, uh, it helps carry away the waste products like carbon dioxide from cells for elimination from the body. It helps in hormonal transport, where it delivers hormones to the target organs. Temperature regulation, it also helps to distribute the heat throughout the body. Definitely immune response, where it helps transport white blood cells and other immune components to fight pathogens at the site of infection. Okay. And also to also help transport neurotransmitters as well. Okay. for signal transduction throughout the body. Now, if we look at this, um, again, fishes have one circuit, two-chambered heart. Frogs have two circuit because there are two auricles and three-chambered heart. Turtles and lizards have also two circuits. Sometimes the ventricle looks like is kind of partially divided. So that's where it is sometimes also called as five-chambered heart. Crocodiles also have four-chambered heart. Okay, two ventricles, two auricles. Birds have four-chambered heart. Mammals have four-chambered heart. Now, if we talk about the human heart, if we talk about the human heart, where the blood passes through it twice, it has four chambers, it has two auricles, two ventricles. Now, one part is the pulmonary circulation and one part is the systemic circulation. Pulmonary circulation is the exchange of blood or movement of blood in general between the heart and the lungs. And systemic circulation is the movement of or exchange of blood between the heart and the rest of the body, right? Now, if we look at it one by one, so this is the right auricle or right atrium where the blood returns to heart from the body and it enters the right atrium. And the passage between the right atrium and this right ventricle is controlled by a controlled by a valve here, which is called the atrioventricular valve, which again helps in regulating the flow of blood into the ventricles. Okay. So then the blood enters the right ventricle from the right atrium. Okay. And then blood is pumped from the right ventricle to the lungs. Okay. From here to here. Okay. There again, this particular flow to the from the right ventricle to the pulmonary artery is controlled by this one here. Is controlled by valves here, which are moon-shaped kind of half moon-shaped valves. That's why those are called semilunar valves. Okay. And then finally, it enters the systemic circulation, where blood returns to left atrium from the lungs. Okay, this is so from the body into the right atrium and right ventricle, this is deoxygenated blood. Okay, and when this deoxygenated blood goes into the lungs, it gets exchanged. Oxygen is diffused via diffusion. The blood gets purified. Okay, purified as in terms of CO2 gets. I mean, O2 is taken up, CO2 is released, and eventually what happens is, finally or oxygenated blood enters the left auricle and to the left ventricle, which is again controlled by this valve here, this one here, okay, this part here, which is called again the atrioventricular valve. The blood flow is regulated at this point from the left atrium to the left ventricle, and then blood is pumped from the left ventricle to, to the rest of the body, okay, via this one, via the aorta, okay. So all the deoxygenated or impure blood comes from the body into the right atrium via the superior vena cava, and then pure blood or oxygenated blood is pumped from the left ventricle via the aorta to the rest of the body. Okay. Now, if we look at the blood pressure changes during the cardiac cycle. So, first is the ventricular systole, where the ventricles get like contracts, and ventricular diastole where ventricles relax. Okay. So what happens is the very first thing, aortic valve opens. Okay. At this point, and then uh, if you look at this particular line here, okay, aortic valve opens, then aortic valve closes. Then if we talk simply about the aortic pressure here, this part here, this part, okay, where it is kind of pumping out pure blood or oxygenated blood from the heart to the rest of the body. Okay. So this is if we talk about the aortic pressure here, first the aortic valve opens, suddenly the pressure increases, and then Once it closes, there is a slight decrease in the overall pressure. And then again, uh, if we talk about this part here, where atrioventricular valves close, atrioventricular valve helping regulating the blood flow from the auricles to the ventricles, whether it is from the right auricle to right ventricle or left auricle to left ventricle. Okay, so atrioventricular valves close, aortic valves open, right? And then when aortic valves close, then the atrioventricular valves open. And so this is the atrial pressure, this is the aortic pressure. This is how the whole pressure graph looks like in the span of seconds. Okay. Now there is something called sinoatrial node, which activates the atria, and electric activity is detected in the atria, and it actually acts as a pacemaker. It actually stimulates the beating of the heart. Okay. Then it helps the atrium or auricles release the blood inside into the ventricles. Okay. And then there's again electrical activity in the ventricles via atrioventricular node. And eventually, when they contract upon the activity of AV node, and finally, when they relax, you can see the recovery of ventricles here, where it has kind of passed the blood to the respective, to either the lungs either the lungs, this part here via this pulmonary artery, or via the aorta to the rest of the body. Okay. Now we know about blood pressure, where it's 120 by 80. Uh, in a it is considered 120 by 80 is considered the optimum blood pressure in a healthy individual, where the upper numerator represents the systolic blood pressure, which is the number of times when the pressure of blood when the heart is actually contracting, and the denominator represents the diastolic number when the heart is relaxing. Basically, how much pressure is exerted on the walls of the arteries. Okay. Now, if we talk about the components of blood, uh, it like in one of the early tutorials, um, it like we read about centrifugation, right? So eventually, centrifugation helps in kind of uh, separating things according to their molecular weight, or even density centrifugations help in separating different components in a sample according to their densities, right? So similarly, blood can also be centrifuged at different speeds and time points for different time duration, and to separate out the different components. So final, like when we really centrifuge blood sample, we finally get three or four layers rather, uh, like distinct layers, like very broad distinct layers are three, and in between at the interface we do have certain like platelets as well. However, it is mixed with the white blood cells. So mostly, majorly, it is three distinct layers, where the top layer, which comprises of 55% of the blood's liquid component, is the plasma. Plasma contains water, proteins, blood gases, sugars, vitamins, minerals, and waste products. And red blood cells are the ones that settle at the bottom, complete bottom, and it comprises about 45% of the blood volume. Okay. And then comes the white blood cells, which comprises of 1% of the blood volume, and then platelets, which are platelets help in again clotting, blood clotting and stuff. Uh, so this is what it looks like after centrifugation in a centrifuge tube when we centrifuge blood. Now, when we talk about red blood cells, RBCs lose their nucleus at maturity. These are denucleated cells, and it makes up about 99% of the blood cellular component here. And red color is due to the presence of this pigment called hemoglobin. Okay. Now, when we talk about the structure of hemoglobin, it is a tetrameric compound. Tetrameric as in uh, and it is a heterodimer also, because it has four domains, nicely folded domains, and where two uh, as in two domains are of the same type, where two are like alpha subunits and two are like beta subunits, nicely folded beta subunits and nicely folded alpha subunits. And similar, as in similar polypeptide chains, these individual are called, and they are named as alpha globin chains and beta globin chains. And to each of the four chains is attached heme, this one here. This is the heme group here, which is a complex of iron in ferrous form and protoporphyrin. So 96% of hemoglobin present in adults is called HbA, and it has two alpha globin chains, these two here, and two beta globin chains, these two here. Okay, alpha 2 and beta 2. Now, if we talk about the lifespan of RBCs, it's approximately 4 months, and iron from hemoglobin present in this heme group is actually recycled in the liver and spleen. Okay. And there is this hormone called erythropoietin, which is actually produced by the kidneys. It helps in the production of RBCs in the red bone marrow. Okay. So what happens in general is erythropoietin production by kidney. Erythropoietin production by kidneys. When there is oxygen deficiency, it stimulates erythropoietin production by kidneys, which in turn stimulates red blood cell production in the bone marrow. Eventually, higher red blood cell production helps restore the oxygen levels, and upon restoring the oxygen levels in the body, or in the blood rather, the erythropoietin production is inhibited. Okay, so this loop goes on based on how much oxygen is available to the body, or how much oxygen is present in the blood. Now, what are platelets? Platelets are actually small colorless cell fragments that play their major role is to help in the process of blood clotting. Okay, these are actually cell fragments basically. So whenever a blood vessel is injured, platelets rush to the site and form a plug to stop bleeding. And how does it take place? How does clotting take place? This is via chemicals that attract more platelets and promote the formation of a stable plug or stable clot. Additionally, platelets help repair damaged blood vessels by releasing growth factors that stimulate the growth of new cells. And if platelets weren't there, even minor injuries could cause life-threatening bleeding. Okay. And when we talk about the source of platelets, platelets are derived from megakaryocytes because they lack, now platelets also lack nucleus, and they have a very short lifespan, usually for about 10 days. Now, if you look at blood clotting, how it takes place. So, the very first thing is endothelium of the vessel is damaged and it exposes the connective tissue. Eventually, platelets come and stick onto it. Okay. So, if you look at this cross section here, these are the collagen fibers, these ones. Okay. And this is the platelet. This violet purple one is the platelet. Platelet releases chemicals that make nearby platelets sticky, and clotting factors from platelets. There are damaged cells also. There's plasma in general, which carries calcium, vitamin K, etc. So eventually, these clotting factors help in activating prothrombin into thrombin. Okay, and upon activation of thrombin, thrombin acts on fibrinogen to form fibrin, and eventually, like all these platelets already were releasing chemicals that eventually make the nearby platelets sticky and thereby forming a little plug. And eventually, when these different factors act on the different, you know, different kinds of clotting factors in the cascade, blood clotting cascade, then fibrin is formed, which eventually forms a fibrin clot here. So you can see this is how a clot, a blood clot looks like. This is the actual image of a blood clot, where these are the red blood cells and these are the mesh of the fibrin clot formed upon clotting. Okay. So the very first thing that is happening is endothelium of vessel is damaged. connective tissue gets exposed, platelets release certain chemicals that make the nearby platelets sticky. Eventually, platelet plug is formed, and also the released chemicals from platelets, damaged cells, and even plasma helps in activating the blood clotting cascade and thereby forming fibrin. Fibrin clot is formed, and the platelets form a plug. Fibrin clot is formed, and the seal is, I mean, the opening or the wound is sealed. Okay. So, in general, why are platelets special? We have looked at blood donation, right? So sometimes uh, RBCs and stuff are always taken and separated, stuff like that. However, platelets are also very much useful and really required, and often is most often needed for patients experiencing chronic diseases such as cancer patients and those awaiting surgeries and transplants. Everyday patients. So this is actually like completely taken from a report from Texas. Okay. So there they report that they say that everyday patients in their community receive platelet transfusions as they navigate their treatments and receive life-saving care. Because platelets expire only 7 days after donation, those patients are relying on special group of donors to meet the daily platelet transfusion needs for their community. So the demand for platelet transfusions has grown dramatically in past 5 years. So based on the data from Central Texas, since 2018, the need for platelet transfusions has grown to 42%. And platelet donation is the most efficient method for collecting platelets, with a single donation appointment potentially saving multiple lives. Okay. Now, when we talk about the immune system, we have gone through this part, like in the previous tutorial also. Still, because white blood cells also comprise, so still white blood cells also comprise, like constitute almost 1% of blood. So we will again look at the immune system and the cells therein, white blood cells and different kinds of um, lymphocytes and stuff which comprise the immune system. So immune system is a complex group of defense responses found in humans and other advanced vertebrates that helps repel disease-causing organisms or pathogens. Immunity from disease is actually conferred by two cooperative defense systems. One is the innate immune specific, like innate immunity, which is non-specific, and adaptive or acquired immunity, which is more specific. Okay. So the very first line of defense, if we call like the barriers, one is the skin, which is a major barrier, which helps in preventing germs from entering our bodies. And then there's a second line of defense where the cells involved are called general attackers. One is definitely phagocytes, which are special white blood cells that act like scouts. They find and destroy invading germs. Then there can be inflammation and fever. So whenever our body is attacked by any kind of pathogen, responses like inflammation and fever get triggered. And fever, for example, can make it harder for some bacteria to grow. So these are part of the innate or natural immunity that we are born with, and they do not create any lasting protection. Then comes the third line of defense, where there are specific attackers. The cells involved are like really specific to the type of pathogen that has entered our body. And eventually, those cells can help in creating memory cells as well. Okay. So one is lymphocytes, another type of white blood cells, where some lymphocytes called B cells help in producing the antibodies. And antibodies are like targeted missiles that attach to specific germs, marking them for destruction. Then there are memory cells. After fighting off a germ, some lymphocytes become memory cells, and these cells remember the specific germ. So if it tries to invade again, our immune system can actually fight it off in a much faster and more effective way. And eventually, the formation of memory cells and the subsequent defense by these cells is called acquired immunity. And when we talk about the innate immune response, so if pathogens pass the physical and chemical barriers, they encounter cells that destroy foreign substances. And these cells are non-specific effector cells of the innate immune system. They include scavenger cells, which can attack pathogens directly, and natural killer cells, which can destroy or destroy the infected or abnormal body cells. Some immune cells eliminate pathogens through phagocytosis by engulfing and digesting them. And then there are innate immune cells that interact with the acquired immune system to fight infections. So if we talk about the cells which can attack pathogens directly, which are the scavenger cells, these are found in most animals and mainly consists of definitely white blood cells, which are also called leukocytes. These are again of two main types, macrophages, big eaters, and microphages, little eaters. So these macrophages are also called as granulocytes, and then there are big eaters, mainly based on the sizes of these cells, which are also called macrophages. So if we talk about the little eaters or macrophages, which are called as granulocytes, these are also known as polymorphonuclear leukocytes because they have an irregular shaped nucleus. And granulocytes contain granules with digestive enzymes and bacteria killing proteins. And further classification of granulocytes can be of three types: neutrophils, which is most common, comprising of 60 to 70% of the total white blood cell count in the body. It engulfs and destroys bacteria. And there are eosinophils, which are effective against really large parasites. Then there are basophils, which release histamine and heparin, and which are important in allergic reactions. Then there are mast cells, which are similar to basophils and help in immune response. So these are produced in the bone marrow and live for only few days, and they move towards the infection sites via chemotaxis. And some pathogens evade them using toxins or resistance to digestion, so they need help from specific immunity. So all these slides, especially the immunity part, has actually been taught in the immune system part completely. Okay, so I'm kind of revising it through these slides again. This part where all the mostly the white blood cells are involved in the immune system. And now since we are reading about circulatory system where white blood cells also comprise a part of blood, so we are going through this again. Okay. So when we talk about the big eaters, the macrophages, they develop from monocytes produced in the bone marrow. Monocytes circulate in the blood and then differentiate into macrophages in the tissues. Macrophages are commonly found in the lymph nodes, spleen, and liver, which filter microbes. Macrophages actually have a longer shelf life, or rather longer lifespan than granulocytes. And macrophages can move more slowly and they arrive later at the site of infection. And they also work by engulfing and digesting the pathogens directly. However, they are comparatively slower than the macrophages or granulocytes. And macrophages are really important for activating specific immune responses. Okay. This is a scanning electron micrograph of a macrophage, this one here, attacking a cancer cell. This is these cells, these are the cancer cells. This is getting attacked by um, macrophages here. Okay. This is the one that needs to be destroyed. And these are the macrophages which are targeting this particular blob of cancerous cell here. Okay. Now, if we look at the general mechanisms by which macrophages work. So suppose these are the bacterial cells. Here the green ones, rod-shaped bacteria, suppose. So macrophages move towards the bacteria and they use their false feet, long thin pseudopods, to reach out to the bacterial cells. And then they engulf these bacterial cells inside here, as shown in this part. And then bacteria is again trapped inside a vesicle, this part here. Okay, here, this part. And then bacteria is contained within a vesicle. Then lysosome fuses with the vesicle and releases the phagocytic enzymes here. And then bacteria gets destroyed and digested. And eventually the undigested portions of bacteria are again packed into vesicles or kind of, processed further, and lysosomal like lysosomes can actually again take it inside and digest the things. Now then there are natural killer cells which actually do not attack the pathogen. So uh, microphages and macrophages are scavenger cells which can attack the pathogen directly by phagocytosis. Okay. And then, however, then there are natural killer cells also, but these do not attack the pathogens directly. Instead, they destroy the infected or cancerous body cells rather, and uh, they look similar to lymphocytes, but they contain granules with toxic chemicals. They recognize dividing or abnormal cells without needing specific immunity, and these cells can kill the cells by releasing cytotoxic granules into them. And if we talk about the abundance of natural killer cells, they are the third most abundant lymphocyte after B and T cells. And natural killer cells are also developed, or the source for natural killer cells are hematopoietic stem cells, and eventually these cells mature in the bone marrow and liver. Okay. Now, if we talk about lymphocytes, these enable the immune system to recognize and respond to different kinds of foreign substances. Usually, the inactive and dormant form, they actually exist in inactive and dormant form unless they get stimulated by specific signals. And eventually, these are like really small round cells with really large nucleus, and little amount of cytoplasm is present inside. And they can actually keep on circulating through the blood and move around the body. And so if we talk about the different type of lymphocytes, they can also, kind of circulate throughout the body via the lymphatic system. Now, if we talk about the different positions or different specific locations of the lymphoid tissue in our body, these are the specific areas where lymphoid tissues are located. And then these small sparkling portions are the lymph nodes here, okay, which are again present at different portions like near the knee, near the inguinal region, like at the lower abdomen, in the shoulder, in the armpits. And if we talk about lymphoid tissue here, there are majorly bone marrow, spleen, thymus, lymph nodes, these sparkling portions here. Tonsils are there, and intestinal lining also harbor a lot of lymphocytes. And these keep on moving throughout our body via a network of lymph capillaries, allowing interaction with macrophages, the big eaters, and other immune cells. Now, this is again a scanning electron micrograph of a T cell, a T lymphocyte from the immune system of a healthy person. This is how a T lymphocyte looks like. And as in the features of a T lymphocyte is like this. And there are two major types. One is the T cell, other is the B cell. So T cells develop from the stem cells in bone marrow, but mature in the thymus, and they make up about half of all the lymphocytes found in our body. And again, T cells and B cells are like portions. They comprise of cell-mediated immunity, where they directly attack the infected or abnormal body cells, and they recognize pathogens inside the body cells and help regulate the activity of B cells as well. And now, what are also T cells actually develop in the bone marrow? T cells also develop in the bone marrow. However, T cells actually mature in the thymus gland, whereas B cells mature in the bone marrow. Like they develop and mature, both development and maturation take place in the bone marrow. And B cells actually help in humoral immunity, and they produce antibodies that bind to antigens in the body fluids. And T cells recognize pathogens inside the body cells, whereas B cells actually detect the pathogens outside the cells of the body. Okay. So they actually travel along with the fluid inside the body. So T cell is the cell mediated immunity, and B cells represent comprise of humoral immunity. Now, if you look at the basic structure of an antibody, or which is also called as immunoglobulin, uh, so all antibodies have the same basic Y-shaped structure here. And there are two chains. This is the light chain, okay, and this is the heavy chain here. And all these light chains and heavy chains, they, especially there are specific portions in the heavy and light chains which are specific to certain antigens binding onto it, which are called antigen binding sites. Suppose this is the antigen here, so they recognize specific portions of, like, specific parts of an antigen. Both the light and the heavy chain comprise of those parts here. And if we talk about the overall structure of an antibody, uh, as shown in this picture, it has four polypeptide chains. Okay, two identical heavy chains, this blue one here. Okay, and two identical light chains here. And all these chains are actually linked by disulfide bonds. And if we talked about the antigen binding site also, which are located at the tip of the two arms of this Y shape here, and it is formed by parts of both the heavy and light chains. And if we talk about, like, at least the like each antibody has at least two identical antigen binding sites. Like minimum, there can be more number of antigen binding sites in these regions as well. However, at least there are two antigen binding sites, because in general the different types of immunoglobulins or antibodies found in our body can also exist in, you know, oligomeric forms. So eventually the whole group can comprise of, like, at least one antibody. One Y shape has two antigen binding sites. Sometimes some antibodies or some immunoglobulin exist as pentamer. Okay. So there will be 10 antigen binding sites altogether. Okay. So, and basically the portion of the antigen that gets recognized by the antibodies here, those portions or those regions are actually called as epitopes. Okay. So epitopes are basically the portions of the antigen which get recognized by the antibody. And this recognition actually occurs through weak chemical bonds. Hmm. Now, when we talk about the variable and constant regions here, again, this is again the same shape, the Y shape only. Here, so again, there are variable and constant regions. So variable region uh, varies between antibodies and determines antigen specificity. Whereas the constant region, this is the part, this constant region of light chain and heavy chain here. Okay, this constant region is similar within the antibodies of the same class. Okay? And this folded shape of each of the domains are maintained by disulfide linkages here, as can be here in between the chains, and even like on one particular domain of a light chain, like in the constant region or even the variable region, we can see there are disulfide linkages, and even two chains are also, like, there's intra-chain as well as interchain disulfide linkages, right? And the tail of the antibody consists mainly of constant regions of heavy chains. So this is kind of reiterating the same thing, where antibody chains are folded into functional units called domains. Light chain, where has one variable domain and one constant domain, this part VL and CL. And heavy chain has one variable domain VH and three or four constant domains like CH1, CH2, CH3 here. Okay. And these constant domains in the tail determine the antibody's biological functions after binding an antigen. Now, if we talk about the disorders, like very commonly found disorders in the circulatory system, one is high blood pressure, which is also called as hypertension. Uh, there can be multiple causes: stress, inactivity, diet issues. And what actually happens is it can eventually lead to different other kinds of heart diseases and possibly heart failure. Then there can be hardening of the arteries as well, which is called arteriosclerosis. Again, whenever mostly it is caused due to high cholesterol and high fat content in the food that you regularly intake. And what happens in arteriosclerosis is the arteries, the thick walled blood vessels, become inelastic, which can reduce the amount of blood flow in the like through them, and eventually this can lead to heart attack or stroke. Then there's atherosclerosis, where the arteries, so arteriosclerosis where the walls become inelastic and walls of the arteries get hardened. Whereas in atherosclerosis, due to deposition of fat, the lumen of the cavity of the artery, cavity of the tube, blood vessel gets narrow. Okay, mostly due to fat deposition within the walls of the arteries. So it again narrowing of the arteries reduces the blood flow to the heart and brain, which eventually can be a cause for heart attack or stroke. Now, when we talk about stroke, it is loss of blood flow to the brain tissue causing cell death, and effect can be loss of brain function or loss of motor control, eventually leading to paralysis, or it can directly lead to, in extreme cases, it can lead to death. There can be coronary blockage as well, a blockage in the coronary arteries of the heart. Uh, the immediate effect can be heart attack or death. Now, when we talk about the locomotor system or the musculoskeletal system, so after the circulatory system, uh, this helps in, you know, helps in movement, stability, helps in providing particular shape and support to our body. And musculoskeletal system can broadly be divided into muscular system and skeletal system. Muscular system includes all types of muscles in the body. Skeletal muscles, in particular, are the ones that act on the joints to produce movements. And besides muscles, the muscular system contains tendons which attach muscles to the bones. And then there's the skeletal system. Main component is the bone, where it acts or articulates with each other to form different kinds of joints, providing our bodies with a hardcore yet mobile skeleton. And integrity and function of the bones and joints is really supported by, there are accessory structures called cartilage, ligaments, and bursae. And besides the main function to provide the body with stability and mobility, the musculoskeletal system also has different kinds of functions, where the skeletal part plays an important role in other homeostatic functions like mineral storage, like calcium, and hematopoiesis, uh, uh, where while the muscular system stores the majority of the body's carbohydrates in the form of glycogen. Okay, so very briefly, uh, this is the system which provides the body with movement, stability, shape, and support. Components are the muscular system, comprising of skeletal muscles and tendons. Skeletal system, bones, joints, associated tissues, which can be cartilage, ligaments, joint capsule, and bursae. Functions, uh, muscles, movement, joint stabilization, maintaining posture, body heat production. Bones, mechanical basis for movements, providing framework for the body. Vital organs are protected. It, bones definitely are the source for blood cell production, and even they help in the storage of minerals. Now, if we talk about different types of muscles that are found in our body, one is the skeletal muscle, which are associated with the skeletal components. And if we look under the microscope, they are they look as striped. Okay, so those are also called as striated or striped muscles, and they help in regulating the voluntary movement of the body, and mainly it helps in locomotion or movement, and also helps in regulating or modulating the body shape. And there are visceral muscles which are present in the inner walls of hollow visceral organs of the body, like the food pipe, reproductive tract, etc. And unlike the skeletal muscles, visceral muscles are actually smooth in appearance, and they are involuntary in nature, and they do not have striations as we see in skeletal muscles. Uh, Again, if we talk about examples of visceral muscles, which are present in the food pipe, helping in peristalsis, and eventually helping in food transport and gamete transport through the reproductive tract. Then there are cardiac muscles also, where the cells assemble in a branching pattern. So, in general, all visceral muscles do not have any branches, whereas cardiac muscles are heavily branched. And like skeletal muscles, they also have stripes. They are also striated. And however, unlike skeletal muscles, cardiac muscles are involuntary, and uh, the nervous system cannot control the cardiac muscles directly. If we look at the figure here, when we talk about the structure of the skeletal muscle, these are this one here, okay, this part here. These are really long cylindrical and multiple nuclei are present here, multinucleated cells. And if we look at the organization, level of organization inside, there's epimysium, the outermost layer, followed by the perimysium, followed by the innermost layer, which is the endomysium. Okay. So epimysium separates one muscle from the other. Okay. That is the outermost layer. And then there are collagen fibers of the epimysium, this part here, which are wavy in appearance. And there's a perimysium also, which is the next layer after epimysium, which like each perimysium covers approximately 100 to 150 muscle fibers. So these individual circles here, um, so these are eventually covered or kind of wrapped around by the perimysium here. Okay, and these individual muscle fibers are like each one. So this one is covered by perimysium, and this individual one group of muscle fibers covered by or wrapped around by perimysium is called a fascicle. All right, and the interstitial space, or as in the space between the cells and or between the muscle fibers, is around 1 micrometer only, which helps in the development of the tunnel in the perimysium, and hence the arteries, veins, and nerves can pass through it here in these parts. Okay, and in perimysium, the one that is covering this fascicle, like covering this group of muscle fibers here, there are collagen fibers that are arranged in wavy forms with crosslinks, which helps in improving the strength and stability of the muscle fibers. Then finally, the innermost layer comes into picture, which is the endomysium. It has very loose connective tissues, and they additionally add up to the strength of the muscle fibers. And it also has links with the perimysium here, as it can be seen in this diagram. Okay. Now we come to how muscles really contract with the help of different kinds of fibers. Okay, and filaments. So muscle fibers are actually lined by the plasma membrane. In this context, it is called as sarcolemma, enclosing the sarcoplasm. The muscle fibers contain multiple nucleus. These are multinucleated. Is also known as syncytium, and the sarcoplasm contains relatively higher amount of glycogen granules and myoglobin. And higher amount of calcium is also stored in the endoplasmic reticulum of the muscle fibers. And it is kind of arranged parallelly, where even the filaments are called as myofilaments in the sarcoplasm, and it is one of the characteristic nature. The parallel arrangement of these myofilaments is one of the characteristic nature of the muscle fiber. Okay, and there are two types of myofilaments actually. One is the actin, and another is the myosin. So, based on the thickness, myofibril can be termed as thin and thick filament, which is made up of actin and myosin filaments. The thinner ones are made up of actin myofilaments, and the thicker myofilament is made up of myosin filament. So, if we talk about the actin filament, which is now when we talk about the contractile unit of a muscle, okay, so there are certain zones where certain, like after contraction or relaxation, these zones can actually reduce in thickness or expand in thickness. Okay, so accordingly, there are actin filaments, which are also known as I band or isoelectric band, whereas myosin filament is termed as A band or anisotropic bands. These are actually arranged alternatively and run across the muscle fibers. And so there are certain portions, certain regions, uh, which is the contractile unit, which is present between, like the Z line and M line, which are fibers with elastic in nature, which are elastic in nature, are present in between I band and A band respectively. The portion between uh, in between two A bands is termed as sarcomere. Two A bands as in um, the anisotropic bands formed by the myosin filaments are one sarcomeric unit, okay, which is responsible for contraction. And the middle line is the Z line and M lines respectively of the I bands and A bands. Okay. Now, if we talk about the actin filament, which is the thin filament, it is one of the major muscle proteins. It is polymeric in nature. What is the monomeric unit is the G actin in case of actin filaments, and it is globular in nature. And the individual G actin monomer weighs around 43 kDa, and the number of actin monomeric units, like multiple G actin, can actually polymerize to form a multimeric fibrous F actin or filamentous actin. Okay. And when we talk about these two filamentous actins, this orange one and the blue one here, these two F actins can coil around each other to form an alpha helix. Okay. And eventually, the thickness of the F actin, the filamentous actin formed by these two individual actin filaments, polymerize like actin polymers, uh, the thickness is around 6 to 7 nanometers, and there are approximately, in general, 14 G actin monomers in one turn. Okay, and eventually, these are actually associated with tropomyosin and troponin, and uh, tro- this is the filament which is the tropomyosin filament, which wraps the like active binding sites on the actin where the myosin can come and bind onto it. Okay. Now, if we talk about myosin, which is another major muscle protein, it is hexameric in nature. Again, if we talk simply, so actin we looked at, G actin is the monomeric form, and uh, polymerized filamentous actin, two filamentous actin can coil onto each other, around each other to form an alpha helix, right? And then there are tropomyosin and troponin, which actually kind of wraps around these filamentous actin to mask the myosin binding sites on actin filaments. Now, when we talk about myosin, myosin is basically, if you like, it has a tail portion and a head portion, and it is hexameric in nature, consisting of two heavy chains and four light chains. These, uh, these are the this is the heavy chain part. These are the four light chain part, and the heavy chain weighs around 200 kDa, whereas the light chains weigh around 15 to 27 kDa respectively. And heavy chain actually forms dimeric filaments. This is the one here, by coiling alpha helically each like onto each other except the N-terminal portion. And what happens in the N-terminal portion is actually globular in nature, and it serves the binding site for other light chains as well as F actin. Now, N-terminal of myosin has actually ATPase activity, and the C-terminal part of myosin, actually this part, C-terminal part uh, of myosin helps in like forms the H zone, whereas the N-terminal part meets together at the margins of A band and it shows ATPase activity. Now we looked at these fibers here, these filaments here, these pink purplish colored filaments here, which are like the tropomyosin filaments. Tropomyosin is a rod-like fibrous molecule. Its molecular weight is around 66 kDa, made up of two different alpha and beta peptides, and both are alpha helical in nature. Double-stranded tropomyosin run parallel with thin filaments in the grooves of the F-actin strands. Then there's troponin, which is a globular protein. It exists as a three-connected protein system. This one here, TPT, like troponin T, troponin I, troponin C. So TPT or troponin T is tropomyosin binding protein, and TPC is calcium binding protein, and TPI is actin and troponin C binding protein. So troponin lie on thin filaments with an interval of 38.5 nanometers. However, the rest is attached with the F actin, the filamentous actin form, and the tropomyosin. So polymerization of desmin results in the intermediate filaments, which is predominantly present in the Z line. And now, in general, what happens at the neuromuscular junction? So, the contraction of muscles is actually initiated upon receiving a signal via neurotransmitters. So, what actually exactly happens, how it is brought about? Let us look at the individual steps. So, very first thing is there are nerve endings, okay, innervation near the muscles, and so this is the axon terminal. Like this is a neuron ending. The axon comes and it ends here. There is the synaptic cleft here. So there are acetylcholine trapped in the vesicles at this portion. Okay. Now the acetylcholine is released from the axon terminal, the endings of the terminal of a motor neuron, and it binds to the receptor in the motor end plate. This is the part. Okay. Uh, and eventually the binding of the acetylcholine helps in igniting uh, triggering or igniting an action potential in the muscle cell. Okay, so this is the neuromuscular junction where it is comprised of the axon terminal and the motor end plate here. So the action potential which is triggered helps, like it propagates through the sarcolemma and down the T tubules here, runs down the T tubules here, and then the action potential eventually triggers the release of calcium from the endoplasmic reticulum, or rather the sarcoplasmic reticulum, which is a storage house for calcium ions. Eventually, these calcium ions are released and it binds to troponin. We looked at troponin C, which is calcium binding protein. And hence, upon binding of calcium onto troponin C, the masked areas on the actin, myosin binding sites, get exposed. And eventually, what happens is cross bridge cycle is formed where the muscle fiber contracts, and eventually calcium is actively transported back into the lumen of the endoplasmic or sarcoplasmic reticulum following the action potential, and tropomyosin blocks the myosin binding sites again and the muscle fiber can relax. Okay. Now, if we talk about visceral muscles are smooth muscles which are kind of spindle shaped or fusiform in shape. They are present mostly like they are mostly present free in nature, for like that is in a single fiber form. It has great elasticity than the muscle fibers. Contractility and extensibility are also higher than the muscle fibers. So there are myosin, actin, troponin, tropomyosin, calcisporin are some of the proteins which are present in the smooth muscles. Then we looked at the neuromuscular junction in the smooth muscle. Um, we looked at the neuromuscular junction in the motor muscle earlier. Now, this is in case of neuromuscular junction in smooth muscle. What happens is the branches of autonomous nerve fibers innervate the smooth muscle in a diffuse manner. The junctions are not connected directly with muscle, but they secrete the neurotransmitters at external space of muscle fiber. Secreted neurotransmitters diffuse through the muscle fiber membrane to transmit the signal. Now, if we talk about the heart muscles, those are heavily branched, and again they have stripes or striated. They are striated, and cardiac muscles are separated by a dark area which are called as intercalated discs. This portion here. Like if we completely cross section this part here are the intercalated discs. This region here. And interconnected cardiac cells are called syncytium. Again, it has more than one nucleus. Heart consists of two syncytium known as the atrial syncytium and ventricular syncytium. So general, what we were talking about at that point, this is the H zone, this is the I band, this is the I band like isotropic band, this is the H zone, and eventually these are the Z discs. This is the M line, like completely the middle portion. Okay. So in response to signals, action potential generated from the brain reaches the muscle cells through neurons, right? At the end of the axon terminal, uh, like it secretes a small amount of neurotransmitter acetylcholine. Acetylcholine eventually diffuses from the membrane receptors, which are present on the muscle membrane to transmit the signal. And eventually, when acetylcholine binds, it opens the sodium channels, resulting in the influx of sodium inside the cells. And upon sodium influx, an action potential in the muscle fiber is generated and it travels along the muscle cell membrane. Okay. And eventually, this action potential helps in depolarizing the muscle membrane, which results in the release of calcium ions from endoplasmic or sarcoplasmic reticulum to the sarcoplasma. Sarcoplasm, actually. This should be sarcoplasm. So, calcium mediated attraction in between the actin and myosin lead to sliding alongside each other. This sliding movement results in muscle contraction. All right. Now, after a fraction of a second, calcium pump actively transfer the calcium from sarcoplasm to sarcoplasmic reticulum, and this removal eventually results in muscle relaxation. So this is how it has been pictorially represented here. So this is muscle contraction is actually explained by the sliding filament theory, which was proposed by Huxley, Niederberg, Huxley, and Hansen in 1954. So what happens is it is a cyclic repetitive process, this sliding filament theory. In general, muscle contraction is a cyclic repetitive process where the actin filaments slide over the myosin filaments and eventually generate a tension in the muscle. Okay. So if we again look at it, so action potential from the CNS reaches the neuromuscular junction. Acetylcholine is released here. Okay. Nerve signals generated action potential initiates muscle contraction, right? So acetylcholine diffuses via the synapse where it binds the receptors, and after binding, receptors get activated, eventually leading to sodium potassium channels and efflux and potassium sodium influx and potassium efflux from the cell results this movement results in the muscle cell generating an action potential. This is where it is happening, and it leads to depolarization of the inner muscle fibers, right? And eventually what happens upon depolarization, the inner muscle fiber activates calcium dependent, like voltage dependent calcium channels in the sarcoplasmic reticulum, and these channels closely resemble the calcium release channels and activate these start releasing calcium from the endoplasmic or sarcoplasmic reticulum. Now what happens is, upon calcium ion release from the sarcoplasmic reticulum, it binds to troponin C or TPC, which is present in the myosin fibril and modulates the tropomyosin allosterically. So in normal condition, tropomyosin doesn't allow the myosin to bind on actin fibers, right? It masks the myosin binding site. Okay. Now what happens when calcium is actually released from the sarcoplasmic reticulum? There's troponin T or TPT. It undergoes allosteric modification, and troponin loses the affinity over thin filament. Hmm? What happens then is tropomyosin eventually migrates from the actin filament, and immediately the myosin binding sites are kind of exposed, and myosin binds strongly at the exposed area on the actin. Okay, this is what is happening here. Okay, and also again this is an endothermic reaction where the process is energy coupled. So myosin utilizes ATP and cleaves it into ADP and inorganic phosphate. Actin helps to release the inorganic phosphate from the myosin, and overall result is sarcomere, like shortening of the sarcomere or the contractile unit of muscle. So in this state, what happens is distance between the Z bands shorten. This part here, okay. These are the Z discs, right? So these two, the distance between two Z discs actually shortens, eventually leading to the contraction of the muscle. Okay, so in general, this is a kind of a fact where the movement of myosin was observed around 10 to 12 nanometers per power stroke, And availability of ATP increases the power stroke cycles energy generation by rising the ATP. So again, what happens is sarcoplasmic reticulum actively collects the calcium present in the sarcoplasm, and eventually there is decrease in the sarcoplasmic calcium level. So calcium eventually starts to migrate back from the actin filaments, which results in tropomyosin remodeling to the native form, where it eventually again masks the myosin binding site. So the native form of tropomyosin replaces the myosin and binds with the actin or the thin filament. Now, if you look at the muscular system disorders, um, muscle strain is something which is an injury where the muscle fibers can tear as a result of overstretching, and it can be a pulled muscle or a torn muscle. So hamstring strains are prevalent in case of athletes, right? Then there can be tendinitis. Tendinitis is the case when a tendon gets there is inflammation of a tendon, uh, even due to overextension or like using the putting too much strain on the tendons, giving without giving any rest. And mostly these are affected in the regions around ankle, knee, shoulder, and elbow. Then there's carpal tunnel syndrome. Carpal tunnel syndrome, what happens is it is a biomechanical problem in the wrist where the median nerve gets compressed between the carpal bones. And this might be occurring this is caused due to repetitive use of the wrist. Okay. There can be muscular dystrophy, which is in this case, this is a genetic disorder caused by defective proteins in the muscle cells. What happens here is there is a progressive weakness in the skeletal muscles, and eventually the tissues, muscle cells, and tissues die. Then there's myasthenia gravis, which is an autoimmune disorder where the circulating antibodies actually block the acetylcholine receptors that we looked at in the neuromuscular junction, right? So this blockage of acetylcholine receptors eventually leads to muscle weakness, and this is eventually the symptom that we get to see is drooping eyelids, okay, and expanding drooping eyelids and expansion to include overall muscle weakness and fatigue. Then we also have Parkinson's, which is a degenerative disorder of the CNS that mainly affects the muscular system and movement. Uh, like four motor signs and symptoms are considered defining Parkinson's disease: muscle tremor or shaking. Muscle can become rigid also, slowness of movement, and postural instability. All right. Now let us look at some of the questions. So which of the following statements correctly differentiates open and closed circulatory system? So open closed circulatory systems have blood confined to vessels. Open systems have hemolymph bathing the tissues. Correct answer is option C. Then, I have the answer already written here. Still, we will go through the question. Mammals have a double circulatory system. Uh, and the supporting statement is it ensures separation of oxygenated and deoxygenated blood, improving metabolic efficiency. So definitely the reasoning statement is the correct explanation of the assertion that has been given here. So correct answer is option A. Like definitely both the statements are true, and the second statement nicely explains the first statement. Now we have to arrange the following structures in the correct sequence of blood flow during systemic circulation, that is from the heart to the body, rest of the body. So from the heart to the rest of the body. That means we have left ventricle, right atrium, aorta, vena cava. Okay. So then we will eventually have right atrium, left ventricle, aorta, right atrium, left vena cava, left ventricle, aorta. So the correct answer will be option, yeah. First is left at ventricle, then it goes to the aorta, then from the body it comes to the vena cava and then enters the right atrium. Okay. Now, if the atrioventricular node in the human heart fails to function properly, which of the following will most likely occur? Ventricles contract before atria? No, this can't happen. Atria and ventricles contract simultaneously? No, because atrioventricular node mostly regulates and controls the contraction of the ventricles. The ventricular contraction becomes delayed or independent or no electrical activity in the heart. Electrical activity might be there because what if the sinoatrial node is functioning properly, but in case AV node or atrioventricular node isn't functioning properly, the ventricular contraction might become delayed or independent. Okay, which combination correctly matches the organism with the type of circulatory system and the heart structure? So the correct option will be option C here, amphibians closed circulation. Sorry, fish closed circulation, two-chambered heart. Okay. Amphibians have three-chambered heart. So which feature is the most characteristic of arterioles as compared to arteries? So arterioles actually have a lining of smooth muscles, and it helps in regulating like those act as resistance vessels with a layer of smooth muscles and helps in regulating the blood flow and pressure via constriction and like vasoconstriction and dilation. So why do veins have thinner walls than arteries? So the reason is they are because they have blood, the they carry blood from the body to the heart, right? And eventually the blood pressure in the veins is lower. Okay, so veins operate under low pressure and thick muscular walls are really unnecessary. However, for arteries, the walls are really thick and the blood flow in veins mostly rely on valves and skeletal muscle pump instead. Next, again, we have assertion reasoning type of question where the first statement is veins contain valves, and the second um, statement blood in veins flows under low pressure and often against gravity. So the correct answer, I mean, both the statements are correct. And eventually, the second statement is kind of explaining the first statement as well. So why? Because there are valves in veins, which helps in preventing the backflow because they usually work under low pressure and often against gravity. So the valves are really essential in this case due to low pressure and gravity effects. The loud sound of the heart is caused by which type of activity? So it is closing of the atrioventricular valves, which actually regulates or kind of regulates the flow from the auricles to the ventricles. Now, which structure normally sets the pace of the heartbeat? That is the sinoatrial node or SN. It is the natural pacemaker generating spontaneous electrical impulses. Now, which combination is correctly matched? Skeletal muscles are actually voluntary and they do have a lot of stripes. They have striations. Smooth muscles are involuntary actually. Cardiac muscles are involuntary and striated with intercalated discs. Correct answer will be option C. And why does smooth muscle contraction occur more slowly than skeletal muscles? Because smooth muscle contraction depends on um, like this myosin linked phosphorylation, making it slower, but sustained contraction is there in case of smooth muscles. The ATP's activity in muscle contraction is associated with the myosin head, right? And or like where we did see that there are four light chains and two heavy chains. Which statement about actin is correct? It forms thick filaments. No. It binds ATP to generate force. It provides binding sites for myosin. It hydrolyzes ATP. The correct answer is it has binding sites for myosin. And these are actually thin filaments. So in cardiac muscles, calcium ions required for contraction are stored in the sarcoplasmic reticulum, and it can come from both the sarcoplasmic reticulum as well as the extracellular fluid. Okay, which is actually essential for excitation and contraction coupling. Which property distinguishes cardiac muscles from skeletal muscles? Voluntary control. Skeletal muscles are voluntary, cardiac muscles are involuntary. Cardiac muscles have intercalated discs, absence of actin and myosin. Presence of intercalated discs allows electrical coupling and synchronized contraction in the cardiac tissue. And actually, this mostly cardiac muscles are involuntary and skeletal muscles are mostly voluntary. And so correct option could be A and D. Both A and D could be correct options here in this case. Which feature is characteristic of the visceral smooth muscle? Visceral smooth muscles have gap junctions enabling coordinated contractions, which help in slow and coordinated movements like peristalsis in the foot pipe. Next is, unlike skeletal muscles, smooth muscle contraction is primarily regulated by this part, the calcium calmodulin interaction. Smooth muscles actually lack troponin, so calcium binds calmodulin and hence activates the myosin light chain kinase or MLCK pathway, and hence the contraction is brought about by calcium calmodulin interaction. Now, when we talk about the sliding filament theory, during the power stroke, ATP binds to myosin head, ADP and inorganic phosphate are released. ATP and inorganic phosphate are released from myosin, myosin detaches from actin, and actin hydrolyzes ATP. So, which event directly exposes the myosin binding sites on actin during skeletal muscle contraction? So, as soon as like the ATP is hydrolyzed, tropomyosin can move away from the uh, myosin binding sites, and eventually myosin can come and bind to it. Okay, so the correct option is release of this part, release of the hydrolyzed ATP causes the myosin head to pivot, pulling actin. This is the power stroke. And eventually, what exposes myosin binding sites on actin during skeletal muscle contraction? Option is, once calcium uh, released from the sarcoplasmic reticulum comes out and bind to the troponin, which is TPC, the troponin C binding protein, like TPC, which binds to calcium ions. Upon binding of calcium to TPC or troponin C, eventually the tropomyosin, there is change in the conformation and hence it eventually exposes the myosin binding sites on actin. The grip on the actin filament loosens and hence eventually it exposes the myosin binding sites, and this overall process requires the usage of 1 ATP molecule. Okay. All right, so that's it for today's session. We will meet again on next Sunday with, I think, the next part is nervous system. That's it. I will end the meeting for now. Thank you for joining.